And thanks for giving me the opportunity to present my research here. This is a project from my PhD that I did at the University of Vienna. And I will be talking about a method that we've been developing to correct genomic GC bias in metagenomic data for species abundance estimates. So in recent years, there has really been a huge increase in the number of metagenomic sequencing or samples that has been done. Here is some data I took from the MAGNIFY study. They plot, you can see in green, the number of metagenomic samples on a logarithmic y-axis that has been submitted to the European Nucleotide Archive. So we have this huge amount of data from all of these studies now. But as also has been noticed in the recent years is that there are all sorts of biases and covariables that are impacting the sequencing data that we have. So we really need to interpret this with caution. And I want to talk about a specific bias that our method deals with in this talk, and this is the GC bias. So for this, I quickly want to go over how we obtain this metagenomic sequencing reads and data. So we have our data, our microbiome sample from the stool samples or soil, and then we extract and process collectively the DNA of these taxa that are in this sample, which gives us a sequencing library, which we then sequence, and then we, in some way or the other, classify the reads to taxa and obtain abundance profiles from this. And the issue now is that these biochemical reactions that are happening here in this process do not work equally well for all of the sequences. And it seems that GC content is a predictor for how well the chances that a sequence has to be sequenced here. And in addition, a problem that we have is that there are very different ways of how you prepare these libraries, different protocols, different library preparation kits, and so on. So we have developed GuaCAMOLE, which is a tool that is available, and we implemented and developed a new algorithm for it. And basically, we look at we classify the metagenomic reads with GuaCAMOLE2, and then we have for each taxon, we look at a GC distribution of observed reads, and we compare this to a GC distribution of expected reads from the reference genomes, which we get from the GuaCAMOLE2 and Bracken databases. And this gives us a projection of the data of observed to expected reads. And now the output of the GuaCAMOLE algorithm are two things. So first of all, we get from a single sample without any spike in or anything, we can get the efficiencies of the sequencing process across the GC range. So this bias curve, if you will, here on the right. And what GuaCAMOLE outputs also then are abundances of the taxa that are not biased by GC content. If you are interested in how exactly the algorithm works, please read our publication. I give you the link in the end of my talk. So as a first evaluation of GuaCAMOLE, we went to a microbial mock community that has been published. So for this, 19 species were pipetted together in a lab in known amounts. So we do know a ground truth abundance of this community. And then this mock community has been prepared with different library preparation protocols, different number of PCR cycles, and so on. And then each of these sequencing libraries has been sequenced. So now analyzing these sequencing reads with GuaCAMOLE, and first of all, we are looking at the efficiencies of the different sequencing runs and libraries, and we can see that not only quantitatively but also qualitatively, the efficiencies are very different depending on the various library preparation protocols that you use. It seems that some protocols favor a low GC content, some favor a high GC content, and so on. And now if you go on and you estimate the species abundances from these data, each point here represents the average error of the abundance estimates of one sample across all the species, and you can see here the highest errors of these that these available methods make when estimating the abundances are, as you would expect, those where the biases are the strongest. So the colors in the two plots are the same. Now, if we use GuaCAMOLE to estimate the abundances, you can see that it's way better, more stable across the different library preparation protocols, and especially for those where a lot of PCR cycles were used, the bias was usually the strongest, and here you see significant decreases, or the most significant decrease in abundance estimation error. So now coming back to what I talked about in the beginning, that there have been so many studies published, we went on to analyze over 3,000 samples from colorectal cancer studies where stool samples have been sequenced of cancer patients. So they are to analyze the gut microbiome. So we looked at 33 studies that have been used in meta-analysis also, that have been further used to analyze them collectively. And we want to use GuaCAMOLE to see how variable this GC bias is across the different studies, and also how the impact of downstream analysis is, or how the abundance correction is, and how it impacts statistics like the biodiversity. Now here on this plot, you can see the sequencing efficiencies for all of these studies. So each plot here is one study where we averaged the sequencing efficiencies of all of the samples. And you can see quite a large variation depending on the study that you look at. The GC bias is different. And this is also a strength of GuaCAMOLE, that we don't make any prior assumption how this bias looks like, but we directly learn this from the data, and a single sample is enough here. And now we grouped these studies based on their bias curves into four groups here. And now we can look at, for these four groups, we can, for example, look at if you estimate the abundance once with Bracken, which does not take the GC bias into account, and then with GuaCAMOLE, how does it change for these four taxa here? And those four taxa have a relatively extreme GC content. And for example, if you go to Fusobacterium nucleatum, which is a taxa that is known to influence colorectal cancer progression, it has quite a low GC content. You can see that in some studies, when you correct for it, it gets over tenfold, the abundance gets over tenfold higher. In other studies, it's twofold higher. So, but what you can see with this is that it does make an impact on your abundance estimates. And then what we also looked like is how it impacts the alpha diversity that you estimate. And if you impact the alpha diversity, if you estimate the alpha diversity from abundances that have been computed with GuaCAMOLE, you get overall a higher alpha diversity average than this would be the case without bias correction. So this shows again that it has an impact on downstream analysis. So this brings me already to my summary. I wanted to show you how GuaCAMOLE allows for this detection of GC bias without any spike-ins or calibration experiments, just from a single sample. It is very flexible. It deals with the different biases that seem to be present in metagenomic samples without making any assumptions. And then, that also this correction matters and has an impact on downstream analysis. Yes, please try it out. I'm happy to get some feedback. Here is the QR code, also links to a recent publication in Nature Communications. And there's also the link to the GitHub repository. It is implemented as an easy-to-use Python package. And let me know what you think. Thank you for listening.